Okay, so last week you did transformation. As I mentioned, I think I mentioned this last week. I saw your gel and I thought I didn't have as much hope, but then apparently Dr. Lally told me that some of you still get really nice red colony, which is great. So like just to remind you what happened, basically you do chemical transformation, which is done through heat shock, and then you spread them on a plate, right? The LPM arabinos, and then you can see the red colony. Is that correct? Were you able to see the red colony at all?
For my group, we didn't get any red colonies.
Okay, so some group do, some groups don't, right?
Yeah, some groups did, yeah.
Yeah,
Yeah, I was also one of the groups that didn't have any.
Oh, okay. Yeah, but it's very impressive that the fact that someone get something in your group, because when I saw the ligation gel, I was quite pessimistic. But hey, you get something, so that's good. So basically today we're gonna talk about what happened after that, right? So what we've done so far is what we call cloning, molecular cloning. But if you remember, the bigger picture that we're trying to do is once you finish cloning, you want to produce this protein, and then hopefully we can purify the protein and sell it, right? Of course, in this class we use red fluorescent protein because you can see it. It's really, you can have a nice visual cue in terms of like where your protein is, where it's gonna come out, easy to keep track. But then like in the real pharmaceutical company, most of the drugs product are colorless, like insulin, for example, right? Insulin doesn't have any color. Growth hormone has no color, right? A lot of those proteins are colorless, right? So, um, and basically today I'm gonna tell you what happened after, what sort of bridge between the cloning after you're done with the molecular cloning into the production. Well, that is the biomanufacturing. So that's where we're gonna start right now. I'm gonna put this one here. Okay, so let's start with this. Okay, so biomanufacturing is essentially the process by which we use biological system to produce something. Manufacturing, right? Manufacturing using biological system. That's what biomanufacturing is. Generally, biomanufacturing can be categorized into two different processes. One is what we call upstream process. And of course, the other one is, you may guess, the downstream process. Downstream process. What's different between the two? In the upstream process is the actual process to produce your protein. So this is the process where you have your cell, your E. coli have the plasmid with the red fluorescent protein there, right? And then you need to produce a lot more fluorescent protein. What you do in this case is you put that in the flask. You inoculate your cells in the flask, you shake them, and then overnight, and then you induce them or something like that. At the end, hopefully by the next day, your culture produce the protein that you want, right? Become like red color. Okay. So that's the entire thing about the upstream processing. For your class, we're gonna skip the upstream processing, and the reason for that is because usually this upstream processing takes, or the activity takes about at least 3 to 4 hours, right? And your class only 1 hour and a half, so that's we won't be able to do it there. I'm thinking maybe next year we can potentially, someone from your group can inoculate in the morning, and then hopefully by 10 o'clock you're able to induce it. So maybe let's think about that. I'll think of the best idea to do it. But for this time, we're not gonna do that. Now in the in the downstream process, on the other hand, you started out with your culture that already have the protein, right? What you're gonna do is now you're gonna pop the cell open. So if you magnify this one right here, you magnify this, you have a lot of cell that produce your protein, right? A lot of your protein inside the cell. What you're gonna do is now you're gonna pop the cell open, right? This is, let's just call it lyse or cell lysis, something like this. And then the cell, now your product is outside. And in addition to your product, you also have a bunch of junk, right? Your other E. coli junk, I put it in this blue right here, right? Now, the downstream processing basically is the process where you actually try to separate your protein, in this case, red fluorescent protein, from the rest of the junk, right? So some people call this separation process. Some people call it purification, right? So at the end of the day, you try to separate your protein. This one can be done. So this is, I'll just write it here. You undergoes to several separation technique. I cannot write technique to get the protein that you want, right? So I don't know if I've told you about the company local to Los Angeles, Grifol and Takeda. They actually collect blood plasma. Well, first they collect blood donation, and then from that blood donation, they separate the red blood cell from the plasma, from the white blood cell. So they only take the plasma. The way they do it is they separate them by centrifugation, right? So centrifugation is one of the method of separation. Oh, by the way, all this thing, I write it down here because it's very important foundational knowledge, and you can best believe they will be in the second questionnaire, right? So centrifugation is one of the separation technique, right? But when you centrifuge, the heavy particle settle down at the bottom, and then the lighter molecule will be at the top, right? So like that is one way to do it, but there are other way, and the other separation technique, which is more precise, if you will, or more cleaner. You separate more effectively, but it can be more expensive, right? So that is usually a separation technique through what we call column chromatography. And there are several column chromatography technique out there. I just want to name a few. You don't really need to know the detail for Biotech 2, but if you're in Biotech 3, you will be exposed to this a little more. And also, if you planning on majoring in biochemistry, these techniques, you will learn this in your upper division biochemistry class. Okay, there are several column chromatography purification method that you can do. One is the one that we're gonna do in your class tomorrow, and that is what we call hydrophobic interaction chromatography. So this is often abbreviated as HIC, HIC, hydrophobic interaction chromatography. The way it works is basically you mix your protein with some agent, some chemical, which that's the solution that you make long time ago, the ammonium sulfate. Remember that? You mix it at a very high concentration to the point that your protein sort of like I don't know if you know your protein. The protein tend to have that the center is hydrophobic, right? But when you put it in this chemical, what happened is that sort of open up this hydrophobic region, right? So now you expose this hydrophobic region. Now, if you float this protein that already have an exposed hydrophobic region to a hydrophobic column. Hydrophobic means they're afraid of water, they're non-polar. So if you kind of like oil, right? So if you flow this protein that's already opened up a little bit with the hydrophobic region exposed to a hydrophobic column, then this hydrophobic region will bind to your column, and that basically make it stick over there. So basically, the more hydrophobic the protein is, the tighter they bind at the column. Now, luckily for us, the reason why we use hydrophobic interaction column is because your red fluorescent protein is relatively hydrophobic. Okay, that's the reason why we can use this. Okay. And the other one, there's another one called affinity chromatography. In here, in affinity chromatography, basically your protein has some sort of like tag, like molecular tag. It can be his tag, like the one with a bunch of histidine, or it can have some other thing. But whatever that tag is, TAG, it binds to the column. So you're gonna set up a column, and that tag, molecular tag, will bind to the column. It's kind of like you're trying to fish, you know, the fish will go to the bait. It's kind of like that. So that's the affinity chromatography. The third one is called ion exchange. So in here, they basically the purification is based on the electrostatic, the charge of your protein, the overall charge. So I'm not gonna describe this in detail, but your protein at any given condition, they have a slight charge, overall charge. They can be slightly negative or slightly positive charge, depending on what buffer you put them in, right? So, and you just have to adjust. If your protein is slightly negative, you may want to put it in that positive column so they can exchange, if you will. You exchange the ion and your protein bind to that column. But again, I'm not going to go describe this in a, further, because this one tend to be you know, a little bit more complex for the scope of this class. But last but not least is gel filtration. Or some people call it size exclusion. Exclusion chromatography. Some people call it SEC, size exclusion chromatography. They basically separate your protein by the size. Okay. So again, today I'm going to just I just give you the overall what they are, but you don't really need to know how each of them work per se for today. Okay. All right, so what are we gonna do today? Well, I'm gonna show you what has been done to your protein, so you can see what's going on from the colony that you produce from transformation all the way until the sample that you will receive tomorrow. So how does the process work? Well, it starts with the with the upstream processing. So you have your plate with a colony, right? The red colony. Something like this, as you put here, right? So what we do is we take this colony, several colony, we put it in that 3 mil culture, and then we do it overnight. Overnight culture, overnight at 37 degrees Celsius, shaking, okay? So, and then the next morning, so this is, let's just call it day 1, okay? In day 2, so like if you do this at Pierce on Saturday, Sunday, we usually usually have a few students coming to the lab maybe on Friday to just pick colony, put it there, to shake it overnight, so that on Saturday, now you have this culture already growing, right? So what you do is, well, at the same time, they have to make a fresh media. So they also have to make this fresh media in a buffalo flask. So they usually make about 80 ml LB ampicillin in buffalo flask. And the volume of that buffalo flask is 250 ml. So it's usually something like this. Let me just show you how do I stop sharing so you can see me bigger. It's something like this. So let's see, something like this, right? So you can see this flask is special. It's buffalo. You look at this indentation right here, right? So, and notice that the volume is actually the flask is about 250 ml, but we only fill it up to about 80 ml. In general, you do not want you want your culture volume to be about 1/3, no more than 1/3 of your flask volume. Now you may ask me, why is that, right? Well, the reason why is because imagine if you have, well, okay, let me just answer that really quickly, and then you can, I'll sort of like take you to imagination, okay? So the answer is because when you want to express protein, you want your cells to have the maximum aeration. So you want them to get enough oxygen as they produce your protein, right? So make them happy. Now, if you fill your your culture with only 80 ml, which is 1/3 of the flask, you can see there's a lot of space over here, a lot of air, right? Now, when you shake them like this, there's enough air that this culture can take. So there will be enough what we call dissolved oxygen. So there's enough oxygen that can dissolve into your culture, right? And that's for the same reason why we have the baffle flask, right? If your flask is not baffle, it's like completely smooth, that's also fine, but it's not, the aeration is not as optimal, right? Imagine if you shake this in a baffle flask, as the liquid hit the baffle part of the flask, it's gonna splash, and that splashing will increase the aeration, the dissolved oxygen that can get into the solution. And that's the reason why, number one, we use baffle flask. Number two, we never fill it more than 1/3 of the flask volume because of that reason. Now, imagine if you fill it, let's say 250 ml, right? Like this. Imagine if you fill it with, I don't know, half of the volume, which is 125, then you have it up to here, something like this. Then when you shake them, it's not as effective, right? So they cannot really swirl around and get the oxygen inside, right? So that's the reason why you generally don't want to fill it more than 1/3 of your flask size. Okay, so that's very important. Okay, now let's see again. Now what so you have your culture here. What you do now is you want to transfer some of this over there. And the moment you transfer some of the culture, you want to know what is the cell density of this 80 ml flask that you just inoculated, right? And to measure your cell density, we're gonna use spectrophotometer. This is the same spectro that you have that you use to measure your dye, right? Remember that spectrophotometer? Something like this. You put your QFET over there, and there's a light source, right? And then there's a detector over here, right? So in here, you will use your what is called your sterile media as your blank, right? Because your blank is the liquid that tells the machine that there's nothing in it. So that's what we call the background absorption, right? So because our media is in the this type of colored media with yellow color, we want to use that media for your blank, but you just don't want to have the cell, right? And then the moment you put this your cell over there, you cannot take your OT0. So this is why usually if you really follow the SOP that I have over and on canvas, the moment you put your cell there, you want to measure the OT. And the OT that we use is OT 600. And the reason why is because at the wavelength 600, there's a minimum absorption done by your media. So your media absorb very little. This yellow color solution absorb very little at OT 600, which enable bigger range for your absorption, for the absorption that is due to your cell, not due to the media absorbing the light, right? So yeah, so you want to measure the OT 600 at time 0. So we should, if you look at the graph in the SOP, or sorry, the table that I have in the SOP, you have to record the time and time elapse. So the very first one is gonna be 0 time elapse, 0 hour, right? And then you record the actual time, maybe I don't know, 10:20 or something like that, whatever the time for the start of your class, right? And then maybe the OT0, we try to target at about OT 0.05 or something like that, right? So, and then after that, you just let it shake. Now, I don't know if you remember from the previous video where we discussed bacterial growth. When you first put the bacteria there, they don't just start growing right away. They undergo what we call lag phase. There's 4 phase of bacterial growth, right? Lag phase, they don't really double at all. And then after that, they will enter this exponential growth phase, or some people call it log phase, right? And then if you grow them for too long, they enter stationary phase. And if you keep it really there for too long, it's going to enter dead phase, right? So we don't know how long this lag phase is, but generally you want to budget about 1 to 2 hours. And this is why maybe I'm thinking now, maybe the next year we do this, we can have one of you coming in period 0, and then start the culture in the morning, let it shake for maybe like 2 hours. And then when you come back around 10, then, hopefully your culture already come out from that lag phase. And this is the reason why the first couple, after you put your first time 0, you measure the OD about 0.05. We try to target for so that the OD is about 0.05. And after that, you don't take the OD again, maybe until 2 hours from that time. So if you start at 10:20, the next time point you're gonna take the sample is at 12:20 or something like that. And by that time, hopefully your culture already come out of the lag phase. Hopefully you get some, I don't know, 0.15 or something like that. I don't know, right? So the time elapsed over here from 10:20 to 12:20 is 2 hours, right? So, yeah, so that's the one, okay? Now, okay, so that's 12. And then after that, now that you know your culture already come out from the lag phase, now they're in the logarithmic phase, they will double every 30 minutes. And this is where you start taking measurement every 30 minutes, maybe 12:50. That's 2.5 hours. Hopefully, if everything is done correctly, they will be 0.3, right? And you keep doing this measurement until to the point where you are ready to induce. Induce means you add some chemical to make sure that to allow the cell to produce the protein that you want. Okay, so for this, you do it. Maybe you let it grow again. You take a sample again at, I don't know, 1 o'clock. 13:20, that will be 3 hours, and hopefully it's around 0.6. The question is, when should I induce them? When should I add this chemical to tell the cell to express my protein? Now, you probably already remember from last time that, what is that? Got it. And then, if you remember last time, the way you induce the expression of your protein is through the addition of arabinose. Remember this arabinose? When you add arabinose, your cell will produce that red fluorescent protein. So that's the inducer that we'll use. Okay. The question is, when should we add the arabinose, right? And that's a good question. The best cell density to induce the protein expression, it vary depending on the media that you have. So for LP, generally you want to induce them between 0.8 to 1.0. Okay, generally. So there are some media that allow the cell to go into hyperdense type of culture. In that case, you can induce them at OD 1.5, like terrific broth or something like that. But for the one media that we have, you want to keep them around, still in the log phase, somewhere over here. But you want to make sure that your cell have enough density, but not to the point that it's too high that the cells start to, produce toxic waste product, or they run out of food, right? And then they enter stationary phase. You don't want that. So that area, which is I indicate with this black arrow over here, is usually about OD 0.8 to 1.0. Okay. So in this case, if by this 1 o'clock 20 minutes, 1:20, it's 0.6, then you don't want to wait for another 30 minutes, because chances are you may overshoot it, right? Maybe like 1.2, right? So you may want to take maybe, I don't know, at maybe 13:35. So this is just 3 and 2 5 hours, something like that. Hopefully the OT is around 0.8, and in that case, you can start inducing it. Okay. Now you can see this one takes some time, right? If you start in your class time 10:20, you're not gonna induce until toward your last period, right? So this is why it's usually not feasible to do this. But again, maybe we can arrange a time where we can induce it at the, or start the culture at the beginning, and then when you come there, we just measure the OT one more time and see if it's enough to induce. Now, once you add, let's say this is you induce, you add Arapinos here. What we do after that, then you're gonna incubate at 37 overnight. Let it shake, okay? So by the next day, hopefully your culture, so this is day, was it day 3? Yeah, day 2, now day 3. Day 3, you have cells that has your red fluorescent protein, okay? Now, this is also another reason why we don't do this part in your class. Usually what we do is we have some volunteer from the school to come maybe in the weekend. We try to do this, and you learn some of this technique as well. So what we do here is that from this flask, you're gonna spin them down. You're gonna put that in the conical tubes right there, and then centrifuge. Okay, so here's your culture, right? You put your culture here, and then you centrifuge it. And then what happened here is that your cell will be at the bottom. And because your protein is inside your cell, the pellet, your cell pellet, will be red. Yeah, you can see before you spin, the entire culture is red, but after you spin, that pellet is red, but then the media is just regular yellow media, right? So what we do is we toss the media, and then you resuspend. Okay, toss the media. And resuspend. Oh, resuspend the pellet. Now, after that, you want to lyse the pellet, lyse the cell. Now, if you follow the Amgen Biotech experience protocol, there's several ways for you to lyse the cell. If you follow the Amgen Biotech experience protocol, they do what we call chemical lysis. So this is what they do using chemical lysis. They add some sort of like detergent, right? So when you add the detergent, the mild detergent that is, your cell will just pop open. And when your cell pop open, that protein now is out, so you can purify the protein. Now, generally, this chemical lysis is not preferred. So if you actually do this in the company, you will never do chemical lysis, especially for drugs. The reason why is because then it will be difficult for you to separate that reagent, that detergent, from your end product, because it's chemical, it's dissolved there. So it's really, really, really difficult to separate that. And this is why in the biomanufacturing company, usually you never use chemical lysis. The other one that also quite popular is what we call mechanical lysis. There are several ways to do mechanical lysis. Basically, you apply mechanical force to lyse the cell. So one of the thing that I used to do is to pump your cell into like very small, like a path, like a tube path like that, and into very narrow opening, and you put high pressure to go to force them into that narrow tubes, and by going so that the cell will pop open, right? So that's the device called microfluidizer. We do that, or French press or something like that. But in our lab, we use sort of like sound wave to lyse the cell, and the device is what we call sonicator. So the process itself is called sonication. let's just call it this one, sonication. So again, like if you've used the Amgen Biotech experience protocol, you will use that chemical lysis where you just add that chemical and then you dissolve the cell and then you spin it again, and you can see your protein, your supernatant now become red, right? But for us, we use this sonication. It looks like something like this. So let me just here. So this is the sonicator right here. So that's the probe right here, right? So it is connected to a machine. So through this cable, it's connected to a power or a sonication generator, right? And then basically you dip your culture here. You put your tubes over there. You keep it on ice, and then you basically just let this the sonic wave to lyse your cell, right? It's actually gonna create this sort of like this sound, like psh, psh, like that, and that's actually also the reason why we don't do that at your school, because it can be dangerous for your ear, right? This is why we have to wear this when we actually perform the sonication. So you have to wear something like this, and then put the culture over there, and then you start turning that one on. And there's some setting that you have to do. You need to adjust to make sure that you what is that called, your cell lies correctly, and you all the while you have to keep your cell on ice, because those sonic wave is gonna increase the temperature. If you don't put them on ice, it's gonna start boiling and your protein will aggregate, right? That's something you do not want, right? So that's basically what you do with the sonication. Now again, like if in the future, maybe we can have some volunteer to come and perhaps do the process in the at Pierce, So anyway, so at the end, let me share the screen again. So at the end, what's gonna happen here is that after either you do mechanical lysis or chemical lysis, now your cell is, or that pop open, right? So now you have something like this, and then you spin it down again. So here is your cell after you suspend it, sonicated. Now you spin it down again. So what you're gonna do, because now you already lyse the cell, when you spin it down, your cell debris is no longer red. Well, I mean, if your sonication is not effective, then you can see the red cell palette still there. You still see the red color. But if you sonicate it correctly, or if you use chemical lysis, then you will see most of the palette, most of your red fluorescent protein will be in the supernatant. So you will end up getting something like this. So something like this. So that's the supernatant. This is actually done by our student right here. That's actually, we have two. One of them is GFP. So this is green fluorescent protein. It's kind of yellow now, but if you shine UV light through this, it's gonna fluoresce green, really beautiful green. I just don't have UV or blue light with me. So but anyway, so you get something like that. Okay. Now for tomorrow, as I mentioned, we're not gonna do any this upstream processing. What you're gonna start with tomorrow is with that cell lysate. So we will have a cell lysate. So let's say from here, what we do is now we take this one, put it in a separate tubes right here. So that's the one that will come to Reseda tomorrow. Okay. So this come to you. There you go. Okay. So now the question is, what do you need to do for tomorrow, right? Well, here we go. So you're gonna use this cell lysate to perform the hydrophobic interaction chromatography column. That's the downstream processing, right? So basically you receive kits like this. Let me just show you right here. Let me stop sharing. Okay, so here's the a set of collection of the column right here. Ooh, this is no good. Someone's open. This is no good. Okay. Yeah, it's no good. Okay. The previous class did not close this correctly. That happens. Okay. Let's see if I can. My hand is not small enough. Okay. Okay, good. Yeah, one of the cap fall off. Yeah. Okay. So basically you receive something like this, right? Some column like this, right? So you basically, each group just need one of this, right? You notice that this whole thing is a column, right? Or at least the the plastic column. And then within the column, you have this what we call resin, this white resin here. Now remember I showed you there are 4 different purification method, right? Um, uh, and what distinguishes one purification from the next is usually the type of resin you have inside. So this is where let's go back to the drawing board right here. So here's the principle, the general principle of protein separation. Okay, so general, I just put general principle here. So usually here's a column right here. That's the spout coming out, something like this, right? And so this is your resin. So generally in the separation protocol, there are 2 type of component in here. One is what we call stationary phase, which is essentially the one that's not moving, the stationary, right? And there's a mobile phase. The mobile phase is the one that move around, right? So the stationary phase in this case is your resin. So in this case, your resin, and depending on whatever resin you use, that will determine what type of purification you're doing. For us, we use the hydrophobic ion chromatography. So therefore, the resin here is the hydrophobic beads. So I think this is a polymethyl or something like that. I don't really know. Yeah. So that's the stationary phase. So the principle of protein separation is as the following. So the protein is in the big, let's say here's the beaker, right? Your protein, your red protein, for example, here is in a mixture with a bunch of other junk. So here, let's use this blue one here, right? And they're all suspended in your mobile phase. So some people call this mobile phase as buffer, right? You put it in a buffer. And then you just flow it there, right? And then what happened here is that the goal is to have your protein selectively bind tightly to your stationary phase more so than the rest of the junk, if you think of it, right? So in this case, your protein will bind to your stationary phase, while a bunch of other junk protein out there is gonna come out, right? And you can dispose this. Usually, you call this the flow through. Your flow through will get a bunch of protein that you don't need. Okay. So and then hopefully after you several wash, then at the end you get with it's only your protein binds to your stationary phase. That's the hope, right? Will that be always the case? Not necessarily, and this is a part of the R&D, research and development, to see if which condition will be very effective at removing all this junk, so that your protein remain, right? So you have to adjust the concentration of your mobile phase and maybe the pH or something like that. And that takes some time to figure it out. Luckily for you, you don't have to do this because the Amgen Biotech experience has figured it out for you, right? So, and because the purpose of this activity is just to take you through the process, you don't really need to try to reinvent the wheel. Let's just use the formula that we already know working. Okay, so so in this case, again, you're gonna take this column that has the stationary phase right here. Now let me Okay, stop sharing again. So notice this one here, the liquid right here, all the column, they are stored in ethanol, alcohol, right? And the reason for that is because your alcohol is, oh, sorry, your resin is made up of from a carbohydrate, kind of like that. If you don't store it in alcohol or ethanol, then there may be some microbe like mold or fungi. They can grow because they eat those carbohydrate, right? They eat them and then they start growing and your column will clog and so on and so forth. This is why when you store your column for a long time, you want to store them in the ethanol, okay, right here. Now, however, you don't want your protein to be in the ethanol, right? This is why the first time you need to do when you arrive is to get rid of this ethanol and rinse it multiple times with water. Okay, so how do we do that? Well, you have another box like this. This one has, you need to collect, okay. Okay, so you have the cup. This one is to collect the whatever dripping from the column. You have the base, it's all about the base, right? And you have the pole. This is not flagpole, but basically you want to attach the pole at the base right here, right? And then you're gonna put your column over here, okay? And the way we do that is by using this very high-tech method, which is the clip and the rubber band. Okay, so what do you do? Like this. So you want the clip to attach right here. Okay, something like this. Okay. And then you attach your column here, technically like this, right? This one can stand, but what if this one fall out, right? That's no good, right? This is why you have the rubber band. So you want to put this one here and then put the rubber band here. So you hold this column in place. Something like this, okay? Now, of course, you want to make sure that your column is not crooked. You want to point straight down, something like this. And then you're gonna put the collection bucket right down here. So when you open the spout, this one can be collected directly here, okay? That's your collection, right? Now, okay, so I hope now you see that how you assemble that. So let's go back to the thing. Okay. So as I mentioned, your stationary phase currently is in ethanol. So here, that's right there, right? So currently it's in the ethanol. I think it's like 20% ethanol or something like that. So you need to get rid of this ethanol, right? So how do you do that? Well, you want to take out this blue bottom here, the blue cap, just take it out. And then you also take out the cap. See, there's this the blue cap over here, right? So you want to take this one out, right? So you don't need this blue cap. When you're done, you want to cap that one back in. But you know, when you want to use it, take them out and then also take the bottom out. And then now you can open the, you know, after you take this one out, you can open the spout, I guess, the spigot, right? And then let it drip. I'm not doing it right now because I want to keep the ethanol there. So I'm not doing it for you right now. But that's basically what you need to do. And then you need to assign someone to watch that liquid drip into this collection tubes, okay? Now, the trick with any of this column chromatography is you do not want your column to be completely dry. And this is why oftentimes you want to let the liquid drip until about 1 millimeter of, or 1 to 2 millimeter of liquid left before it goes down up into your, into, you know, into the that white resin. So you can see from here, right? Okay, let me unshare that again. Right. You see there's that liquid right there, right? So this one is about 1 centimeter-ish, which is fine, right? And then you want to, when you store it, you want to put about 1 centimeter or something like that. And then you want to drain it until it's about there, about 1 millimeter, maybe about somewhere like that, okay? But it's just barely just 1 millimeter or 2 millimeter above that white resin somewhere over there, okay? Somewhere, yeah, somewhere here, okay? So once you see the liquid is about 1 to 2 millimeter away from the resin, you want to stop it by closing the spigot, okay? Okay, let's go back here again. So now after that, after you take the ethanol out, you want to wash it with water. So wash with water. And the way we wash it with water is simply put about, here's the about 2 column volume of water. Now you may be wondering, what, Dr. K, what is this 2 column volume? Or we usually call this 2 CV. CV means not curriculum vitae, that's column volume. What is this so-called column volume, Dr. K? Well, column volume is the unit of measurement that is used by, you know, biochemists a lot to basically communicate how much mobile phase has gone through the column. The reason why we use column volume is because, you know, it depends on how much volume you use for your resin, right? How much, for example, like this resin about this stick, the column volume is about 1 mil. So you only use 1 mil column volume for this, right? But and therefore, if you just put 2 mil, that's more than enough because it's 2 column volume, right? But if your column volume is 5 mil, the 2 column volume is not enough at all, right? So when you wash it, you want to wash it with more volume because your column volume is larger, 1 mil versus 5 mil. So this is why biochemists usually use this, column volume as a unit to describe how much buffer they flow it through the column, right? So okay, so we usually wash it with 2 column volume of water one time, and then you let it drain, and then you repeat one more time. The reason why we want to do this is because your protein, you will put your protein in the ammonium sulfate, and if you don't wash the ethanol completely, the moment the ammonium sulfate come in contact with your ethanol, they may precipitate, and when they precipitate, they make this solid, the salt, and that will clog your column, and you'll be like one of the students, how come mine doesn't flow? My friend already done, I'm still waiting for this to flow. So yeah, anyway, so make sure you don't do that. So make sure you get rid of the ethanol completely, maybe wash it with 2 column volume, and then you repeat it one more time with 2 column volume. I just put 2 CV here, okay? And then from after you wash it, now you want to equilibrate. I cannot write. Okay. Equilibrate. Equilibrate means you condition your column so that they are ready to receive your protein. So you condition them to be at the same buffer condition, right? So if it's pH 5, pH 7, then it has to be pH 7 as well. If the ammonium sulfate is about 2 molar, this is also 2 molar, and so on and so forth, okay? So when you equilibrate them, you want to use the wash buffer. I think this you're gonna use the solution that you make before, and that is your 2 molar ammonium sulfate. That's your equilibration buffer, right? So remember in your in the fall semester where you are being separated into groups, some of you make 1.3 molar ammonium sulfate, some of you make 2 molar, and some group make 4 molar. Guess what? The 2 molar is the equilibration buffer. So you're gonna use that 2 molar ammonium sulfate. We float them. Just refer to your Amgen Biotech experience protocol. If you remember here, where is that? This one right here. Yeah, this one. Yeah, the 2 column volume distilled water. Yeah, this one. Apply 1 column volume of the column equilibration buffer. By the way, this one can be found over here. If you look at your canvas activity number 8, biomanufacturing, I told you we skip upstream processing, right? But if you want to read it more, you can do it here. The protocol is in here, downstream processing. Okay. So after 2 column volume of distilled water, you apply 1 column volume of CEB. This is your column equilibration buffer, CEB, right? And repeat this 2 more times for a total 3 column volume of equilibration. So again, you put 1 ml, let it drain, and 1 ml again, let it drain, and then the third one, 1 ml, let it drain, okay? And then while someone is doing that, remember this one is gonna take some time. You have to wait it drip, 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 right? While someone is doing that, perform step 22, 23, you can have someone else, someone in your group to obtain the lysate, something like this. So Dr. Lally will have lysate like this. What you're gonna do is you're gonna, well, I don't think you need to spin the sample down. It's already, they already give you the lysate, okay? So you can skip 24, 25. We only give you the lysate already. So take only 0.5 ml of the lysate, only 0.5 half ml. And then, and then you add half ml of the binding buffer. Now, the binding buffer, so here you have your lysate here. Let's see here. Here's your lysate. You have a separate tubes. You have a separate tubes, and then you put half ml of this, and you also add half ml of binding buffer. Binding buffer. So what is this so-called binding buffer? That's your 4 molar ammonium sulfate. If you really think about it, you take half ml and you mix it with half ml. So essentially you're diluting it 1-to-1, which means you dilute the concentration of your ammonium sulfate from 4 into 2 molar. Wait, Dr. K, that number sounds familiar. 2 molar. Well, guess what? Your 2 molar is the same concentration you equilibrate your column with. And this is why you equilibrate your column in the with 2 molar ammonium sulfate, so that when you apply your mobile phase, effective concentration of ammonium sulfate is also 2 molar, because you mix it half mil and half mil, right? So, okay. So from there, and then again, if you follow this here, so okay, so you apply, so you should have about 1 ml of sample at this point, right? And then you apply the sample from step 28 from here to the column and let it drain. So of course you have to, remember your column now, you just put someone in your group already put column equilibration buffer, the 2 molar, right? And then you stop at about 1 to 2 millimeter from the resin. Once that one's done, you want to put that 1 ml of your RF, the cell lysate and the binding buffer into this, about 1 ml, and let it drain. Okay, and all the while you can, if you want, you can collect your flow through. That's, here. If you want to collect your flow through, you may want to put Eppendorf tube, the 1.5 mil microfiche tube, just collect it from the bottom, right? And then from there, you wash it with, wash the column with 2 column volume of wash buffer, 2 column volume, and repeat this 1 more time. So let's just put it here in our diagram. So after this, you apply the sample to the column, and then let it drain, and then from there you wash with 2 column volume of wash buffer. What is this wash buffer? That's your 1.3 molar of ammonium sulfate. Now you hopefully you see now why you have to make 1.3, 2, and 4 molar ammonium sulfate. Okay, so you wash it with 2 column volume of wash buffer and repeat it one more time. If you want to collect your wash, you can do so if you want, right? And then I want you to also pay very special attention to your observation. So when you apply your sample here, if you apply the sample to the column, I want you to make an observation. Where do you see that red fluorescent protein is? Is it at the top? Is it diffused throughout the column? Or is it coming out from the bottom, right? When you apply the wash buffer, again, does your protein stay on top, or is it going through the column, or is it coming out, right? So you want to make that observation, okay? Now, hopefully, after you finish the second time of 2 column volume wash, hopefully all the junk protein already out, so leaving only your red fluorescent protein bound to your stationary phase. Once that happens, you want to proceed with elution. I think the protocol also specify how you do your elution. Right here, add 1 column volume of elution buffer, but make sure you obtain 2 1.5 mil microfiche tube because you want to collect that sample. Okay, you want to add 1 column volume elution buffer and let it drain. You collect the first the first column volume, which is 1 mil, in the 1 mil microfiche tube, and then you do another elution buffer and you collect it in another tubes again. So that's step 32 and 33. Hopefully you got your protein there. Okay. And then once you're done, you have to store or clean up the column and store it back in ethanol. And here's the step. So again, you add 2 more column volume, you let it drain to get rid of all the junk that's still bound to your column. And then you add 5 column volume of water, 5 ml. And then finally you add 2 column volume of ethanol, let it drain the first one, and then the second 2 column volume, you want to leave it about 1 centimeter, just like this. Basically, you want to leave it like this. Once you're done, you want to cap the bottom, cap the top, and then we give it to your instructor so we can put it back here and return it to the Amgen Biotech experience. Okay, and then once you're done, hopefully the next time we meet on Friday, or maybe on Friday or the next week, we can do the analysis of your protein by running SDS page. And I think that's pretty much all I want to tell you today. So I guess maybe I'm not seeing you, but Dr. Lely will see you tomorrow. Have a good evening.
Thank you, Dr. K.
All right.